All right, here we are with meiosis part 2. We left off talking about the fact that gamete cells need to be haploid in nature so that the fertilized egg from both the sperm and the egg have a 23 set collection of chromosomes. Let's go ahead and we're going to talk about the process of interphase. So both mitosis and meiosis, the cells go through interphase in which they grow and in which the DNA is replicated, right, through synthesis, S for synthesis, and in which the cells grow again, getting ready for division. In prophase 1 of meiosis. So when we talk about meiosis, we're going to have two sets of meiosis. We're going to have meiosis 1, which is the part of meiosis in which we are going to get a new and unique combination of genetic material, resulting in offspring that have a lot of genetic diversity. In meiosis 2, we're going to be getting from a diploid cell with 46 chromosomes arranged in 23 pairs down to just 23 chromosomes, which will then allow for the reproduction process. But let's start at the top. Let's talk about prophase 1. In prophase 1, chromosomes form. Now, during this time, there is a crossing over or a recombination of genes. Some of the genes from our biological mother's copy will cross over with the genes from our biological father, and so the resulting chromosomes are a unique mixture of the genetic material. The chromosomes are no longer identical. After that, the nuclear membrane breaks down, and the microtubules, or the spindles, attach to the centromeres of the chromosomes, and we get ready for metaphase. Now, just like in mitosis, in metaphase, the homologous matching pairs, which aren't quite so matching anymore, line up in the center of the cell, of course, attached to the spindles. We're going to move on into anaphase 1. In anaphase 1, the pairs separate, dividing the pairs in half. So we're going to have each new cell is still going to have 23 pairs with 46 chromosomes. But in each of the daughter cells, they're going to be different. They are not going to be matching sets. So now we have two daughter cells with a unique combination of genetic material. The formation of the cells continues through telophase, where the cell divides, a new nucleus forms around those chromosomes, and the cell prepares itself for meiosis 2. Very important as we go into meiosis 2 that there is no interphase between meiosis 1 and meiosis 2. There is no DNA replication. So at this point, we have two daughter cells with 23 pairs of chromosomes, and we are not going to be doubling those up. So we are going to take a full set of DNA, and as we move on to meiosis 2, we're going to be cutting those full sets, right? Those diploid cells down in half. And we're going to do that, again, going through the process of prophase. So starting with prophase, we're going to break down the nuclear envelope. The cells are going to align in the center with the microtubules, the spindles. The difference is that each cell now only has one copy of each chromosome with two chromatids that are unique. So we have two chromatids that are going to have a little bit of different genes from that crossing over in meiosis 1. And we're going to have 23 pairs of those. There is no crossing over, however, in this part of meiosis. In metaphase 2, the chromosomes line up in the center. We have a random alignment, meaning there are a lot of different possible combinations that could happen. In anaphase 2, remember we said we have a chromosome, right, with two chromatids, which are the two copies. They are pulled apart by the spindle fibers. Going into telophase, where each new nucleus forms and cytokinesis happens. This means that we now have 4 cells. Each of these cells is haploid, meaning they only have one half of a chromosome pair, or they have a full set of DNA, but only one copy of the 23 chromosomes that they need. These are our gametes, our sperm cells and egg cells. Haploid, ready